In the previous class, I described the cell, the theories of the cell, the functions of all of the components of the cells, where we talked about mitochondria, the nucleus, the cytoplasm, everything that has to do with the cell. Then we talked about all of the scientists that worked on the theory of the cell. Then after that, I described a whole lot of things you can do in biology. That was the first class. The purpose of this class is to help you to cover the entire syllabus in biology, and this will be the second class of the syllabus. Today we want to now talk about that cell and the environment of the cell. So we want to talk about the cell and its environment. So welcome back to Primer's Classroom. I'm popularly known as Dr. Sodium. So the first thing we'll be talking about is the cell and its environment, the cell membrane. So we described the cell membrane properly again. We talked about it in the previous class, but just to quickly describe it again, we talk about what is the influence of the environment on the cell membrane. Then we'll talk about all of the processes involved in transportation within the cell and outside the cell. Then after that, we'll talk about that nature of the environment that the cell finds itself. Is the nature isotonic? Is the nature hypertonic? Or the nature is hypotonic to the cell? Then if a cell finds itself in any of these environments, what is the influence? That is when we start talking about plasmolysis, haemolysis, turgidity, flaccidity, and the likes. So briefly, let's describe the cell and its environment. Now, we have talked about the cell. Now, what do you mean by environment? Environment is the entire surrounding. Everything that surrounds the cell is called its environment. But why do we have to get bothered about the environment of the cell? Now imagine this. Most cells are living. Now, these living cells, what do I mean? That means they carry out life functions. They have the nucleus that determines a whole lot about the activities of the cell. They have the mitochondria that helps the cell to produce energy. Now, for mitochondria to produce energy, it needs glucose. Where will the glucose come from? It will come from the environment. Now, this glucose will find its way into the cell. Now, for the cell to breathe, needs oxygen. Where will the oxygen come from? The oxygen and the CO2. The oxygen comes from the external environment of the cell. They find its way into the cell. So everything that surrounds the cell is what we call the environment. Now you need to note something. Every cell have to carry out life functions because they are living. So now they need to get all of these things from their environment. So now we talked about unicellular organism. What will be described as the environment of a unicellular organism? You know, it's a unicellular organism. It's an organism that is made of a single cell. Now, that means this organism environment is where it lives. It can find itself in the air, in the soil, in water, which can either be the ocean or the fresh water. If it finds itself in the ocean is more salty. So the organism has to adapt to a salty environment. If it finds itself in fresh water, the fresh water is less salty compared to the ocean. So these are the things we want to talk about. But for multicellular organisms, look at the way I am built up. I am built up from cell. That's what we talked about in the previous class. For the cells in me to function, or for the environment to influence them, most of these cells are surrounded by body fluid. But it's the liquid portion of the body that we have, and these cells are bathed in that body fluid. Now, this body fluid determines a whole lot of things. The fluid you have inside the cell is called the intracellular fluid. Fluid within the cell is called the intracellular fluid, while the fluid outside the cell is called the extracellular fluid. Most materials are suspended in this fluid and they can be exchanged between the cell and its environment. Now, is it everything that is in the environment that will find its way into the cell? Everything in the environment has to be regulated. For both animals and plant cells, we have what we call the cell membrane, and this cell membrane is also called the plasma membrane. Don't forget that the plant cell has what we call the cell wall, and that cell wall determines a whole lot about the plant cells, which I might still describe. So when you're now talking about the cell membrane, as we described in the previous class, is a phospholipid bilayer that is made up of majorly lipids and proteins. Now, these proteins are integral proteins and the peripheral proteins. There are some part of carbohydrates that forms part of the cell membrane. All of this has been described in the previous class. But the most important thing you need to know is that this cell membrane is what regulates what goes in and what comes out of the cell. So now the function of this cell membrane is very important. Oxygen can diffuse through the cell membrane, CO2 can move out of the cell membrane, glucose can come in, and all of these things you talk about. What are now the processes by which things can come into the cell and move out of the cell? You must have heard about diffusion, osmosis, and active transport. Now let's quickly differentiate the two. We have what we call passive transport and we have what we call active transport. Passive transport is the movement of molecules without the use of energy. So the movement of molecules from one region to another without the use of energy is what we call passive transport. While the movement of molecules with the use of energy from one point to another is what we call active transport. Now let me get this clear to you. Just imagine you have a ball from a mountain, then you dropped the ball, then the ball rolls down itself under gravity. But when you have to take that ball up, you might have to input energy. So passive transport is the one that you just drop. So the molecules just move from where it is more to where it is less. But now imagine moving things from where it is less to where it is more. It might take me energy, and that is what we call active transport. So under passive transport, we have what we call diffusion, we have what we call osmosis. Diffusions are of two types. We have what we call simple diffusion and facilitated diffusion. But let's start with diffusion. So if gases are going to move across the membrane, then they go on by diffusion. There are some molecules that are very big enough that can't just diffuse through the membrane. Then they have to pass through channel. Now that is carried by some carrier proteins, and that is facilitated diffusion. So what is diffusion? Diffusion is the movement of molecules from a region of higher concentration to a region of lower concentration, or the movement of molecules across concentration gradients. So just saying that you are moving from higher concentration to a lower concentration. There are factors that can affect diffusion. If you increase temperature, then the rate of diffusion will increase. If you look at the molecule, the size of the molecules also determine. The smaller the molecule, the faster the molecules will move. You understand that? So all of these things can affect the rate of diffusion. Now, where do we need diffusion in the body? You remember, fetus have to feed, and they get oxygen, they get most of the nutrients from the parents by diffusion. So also gases has to be exchanged during respiration. Gaseous exchange also takes place by diffusion. If you spray a perfume from this corner, the perfume will spread throughout the room, and that is also by the process of diffusion. So now when you're talking about facilitated diffusion, I told you the other time there are some molecules that are too big to just move across the membrane. So these molecules must be carried by what we call carrier proteins across the membrane, and that means that the movement is facilitated, is being processed. So let's talk about osmosis. Osmosis is defined as the movement of water molecule from a region of lower concentration of salts to a region of higher concentration, meaning that the salt concentration at the other side is higher. What does that mean? Imagine the two bowl is filled with water and salt, but those yellow dots are the ones that represent the salt. The salt is more on this side, while the salt is less on this side. Now water will move from where the salt is less to where the salt is more, and that is why you will see the water level rises up in that container. So that is what we talk about osmosis, and osmosis takes place across a semi-permeable membrane. So the movement of water molecules from a region of lower concentration to a region of higher concentration across a semi-permeable membrane is what we call osmosis. Now, a whole lot of things can be used as examples of semi-permeable membrane in the laboratory, just like the bladder of an animal, a peeled yam, a peeled pawpaw, cellophane paper, or even tissue paper. Anything that can stop other things from moving and allowing water to move, then we call it, but don't forget, not we call it a semi-permeable membrane. That means that a semi-permeable membrane is selective. Do you understand that? You're talking about semi-permeable membrane. Some are living, some are not living. Like the cellophane paper now is just a paper, it's not living. So don't forget that. While the yam, it's a plant cell that is living. So we can talk about living semi-permeable membrane and the non-living semi-permeable membrane. So when do you have osmosis? The movement of water across the xylem takes place by osmosis. Every time you hear about movement of water majorly, then you talk about osmosis. Now, when water moves into the cell, we call it endosmosis, endoosmosis, meaning that water is moving into the cell. When water moves out of the cell, we call it exoosmosis. Now that you know about osmosis, let's talk about active transport. Active transport is the movement of molecules against concentration gradients. You move ions against concentration gradients. The roots of plants, so the ions at the roots of plants is lower than the ions in the soil, but the ions from the soil still has to move into the roots of plants, meaning that where the ions are more in the soil will come into where the ions are less in the roots of plants. So that takes place by what we call active transport, and energy will be used. So the pumping of potassium ion in the cell too, you will know about all of these things as we move on with the class. Some might be getting too much for you at this level, but just follow through. Now, the movement of those ions from region of higher concentration to region of lower concentration is what we call active transport, and energy is going to be needed during that process. Now that we have talked about the processes through which things can move in and out of the cell, let's briefly talk about the environment the cell can find itself. We have different solutions, 3 solutions. One solution is called isotonic solution, one is called hypertonic, and one is called hypotonic. Don't forget, it is the solution we are describing now, not the cell. Let's get this right. We have an isotonic solution. Isotonic solution is a solution that has equal concentration as the cell. So when you place the cell in such concentration, everything will be balanced. It's an equilibrium process. But when we say hypotonic, hypo means lower. So meaning that if you place a cell in an hypotonic solution, the hypotonic solution means the salt concentration in that solution is less compared to that of the cell. The cell has a higher concentration of salt than that of the solution. So a solution with low concentration of salt compared to that of the cell is called hypotonic solution. While hyper means more. That means the solution has high concentration of salt compared to that of the cell. So we call that hypertonic solution. Now, for this solution that we are talking about, what is now the influence on the cell? When you place a cell in isotonic solution, since the salt concentration is equal, you know, osmosis means that if the salt is more on one side, then the water will move from where the salt is less to where the salt is more. So if you place it in an isotonic solution, the salt is equal, meaning that water will not go into the organism, neither will water leave the body of the organism. Do you understand that? That is isotonic. Hypotonic, when you place a cell in an hypotonic solution, you remember hypotonic solution has less salt concentration compared to the cell. That means water will start moving from the hypotonic solution into the body of the cell. And when the cell start absorbing too much water, what do you think is going to happen? We will talk about that. Let me just finish explaining this. Just get the concept clear. Now, for an hypertonic, you place the cell in an hypertonic solution, meaning that the solution is more concentrated than the body of the cell. And what does that mean? That means that the cell is going to lose water into the environment, and that is called an hypertonic solution. Now, what is the influence of this? If you place a cell in an hypotonic solution by osmosis, the cell is more concentrated than the hypotonic, so water will start moving. Then all of this leads us to some terms that we call plasmolysis. So what is plasmolysis? Plasmolysis is the shrinking of cell. Plasmolysis is used for plants. When you place a plant cell in an hypertonic solution, the plant cell will lose water to the environment, and by losing water to the environment, it will shrink and the plant cell will most likely die. When it leads to the death of the plant cell, we call it plasmolysis. But if you can quickly supply water to the plant cell, then it will be deplasmolized. That means that it will go back to its normal shape. So if you place it in an hypertonic solution, you remember it's going to be losing water because the solution is more concentrated than the solution of the plant. Then it's going to lose more water to the environment, and by losing water, it will be shrinking. The cell will be shrinking, and the plant will die. But the plant cell will die. But eventually, if you supply more water, it will deplasmolize and it will come back to life quickly, not after a very long time. Another thing is that there's what we call flaccidity. Flaccidity is almost the same thing as plasmolysis, but flaccidity does not lead to the death of the cell. So the plant will become flaccid. The cell becomes flaccid, will shrink when it is placed in an hypertonic solution. It will lose water and it will shrink. There's what we call turgidity. Turgidity means turgid. It means strong. Now, turgidity is when you place a plant cell in an hypotonic solution. You remember hypotonic, the water concentration in the hypotonic solution is less than that of the concentration of the cell. So now the cell will start absorbing water from the environment, and by absorbing water from the environment, it will swell up. Let me use that English. That means swelling up means that the cytoplasm will get bigger, and the cell wall will counteract that getting bigger by what we call tug-up pressure, and the cell will get bigger and rigid. And by the time it gets rigid, then it will make the plant to stand so strong and so firm. That takes place in your guard cells too, and that's when guard cells become rigid and turgid. It helps plants to be strong. So turgidity is important, it's positive use. So when a plant cell finds itself in an hypotonic solution, it becomes turgid because it would have taken in a whole lot of water and it will become turgid. Then for animals, we have the red blood cell in plasma. If red blood cell is placed in an hypertonic solution, now what do you think? That means the solution is more concentrated than the red blood cell. Then the red blood cell will lose its water to the solution, and at the end of the day, the red blood cell will shrink, and that process is called crenation. crenation, crenation. The shrinking of the red blood cell is called crenation. Now when you place the red blood cell in an hypotonic solution, that means the concentration of the salts in the cell is more than the concentration of the salts in the solution. So what do you think will happen? The cell will absorb water, will take water from the solution, and it will get bigger and bigger and bigger. And because there is no cell wall, then what will happen? The cell will burst and release its contents into the environment. The bursting of red blood cell is called haemolysis. So haemolysis is the bursting of red blood cell. Plasmolysis is the shrinking of plant cell. Turgidity is the swelling up of the plant cell, while flaccidity is also the shrinking that does not really lead to the death of the plant cell. Why crenation is the shrinking of the red blood cell. So don't forget all of these processes. They are the influences the environment have on the cells. So but this is very important. Understand how ions move from the roots of from the soil to the roots of plants. Understand how oxygens diffuse, how carbon dioxide diffuse within the organisms. For the plant too, carrying out photosynthesis, how CO2 diffuse into the leaves of the plant, how food are translocated into the entire bodies of the plant. So you have to understand all of these things. So then don't forget the function of the cytoplasm. So don't forget the function of the cell membrane, how it regulates what goes in and out of the cell. So briefly, we have talked about the cell and its environment. So the environment is the surroundings that surrounds the cell. We have talked about the processes through which things move in and out of the cell, and that is regulated by the cell membrane or the plasma membrane. So the processes we talked about were the passive transport and the active transport. The passive transport is diffusion and osmosis, while the active transport is the movement of molecules with the use of energy. Then we talked about the different solutions that a cell can find itself, such as an isotonic solution, hypertonic solution, and hypotonic solution. That is what we talked about. Then after that, we talked about plasmolysis, turgidity, haemolysis, and all that has to do with it. So please make sure you watch this video carefully, write the notes you're supposed to write, and if you're here to subscribe to our channel, please make sure you subscribe. Leave a comment about if you have learned anything about this class. I'm going to upload some questions for you to answer now, and make sure you solve them. Then after this, I'll make a video where I will solve all your past questions on these aspects I've taken, the cell and its environment. So pay attention to the video and make sure you subscribe so that you get updates on subsequent videos, and share this with your friends so that they can learn from the comfort of their home. So this is topic 2. We'll be moving on to topic 3, which is classification in biology, where we talk about the monerans, the protistas, the fungi, the animalia, and the plantae, the gymnosperm, angiosperm, and so on. So get ready and make sure you stay tuned. There's a WhatsApp channel and Telegram channel where we're sharing the questions and the contents of the video, please make sure you follow through so that you solve. During the week we practice questions and I explain all of these questions in the video. So I'll see you in the next class. Thank you.